Hello, welcome to Chapter 7, Lecture 6. The topic for today is cytoskeleton, and we're going to talk a little bit about the components of the cytoskeleton and how those components both structure a cell and move the cell. You can see this picture is obviously not a cell, but it is a good picture of a house, and if you were to strip all the siding and all of the paint and all of the shingles and all that stuff off of a house, you would get a house that has only been framed. And I like to look at this in terms of cell cytoskeleton because the cell is the same way. The house has to be framed, the cell has to be framed, and in a house we use lumber, but in a cell we use proteins. And so if we were to strip all the cell membrane and cell wall and organelles and cytoplasm and DNA and all that stuff out of the cell, you would be left with a protein framework that gives the shape and structural integrity to a cell, much like you'd have just lumber giving the structural integrity to the house. Okay, so let's start. The cytoskeleton is really important when we talk about supporting and movement and how the cell supports itself and moves. And so three big ideas for today. One, cytoskeleton is how the cell supports itself, and cilia and flagella are both ways in which the cell is going to move either materials or itself. The cytoskeleton, like I said, is made up of protein, and cilia and flagella are two distinctly different structures. Both of them are extensions of the cytoskeleton on the outside of a cell, but the cilia, which can be seen down here in this actual electron microscope image, that all of these little tiny projections that look like little tiny hairs on the outside of a cell, much like an anemone that you see on like Finding Nemo, these things will beat in one direction, much like a wave around a sporting complex. If you've ever been to like a football game or a basketball game or a baseball game, when crowds are feeling rather frisky and wanting to cheer for their team, they typically will start the wave, or one person will start the wave and it'll carry on, and it beats in one direction all the way around the stadium. That's the cilia, okay, or the cilia acts just like that. And cilia are used to move materials around the cell. The flagella is a single whip-like propeller, and it can be seen up here. Okay, when you have a cell like a sperm cell, these tails are used to actually move the cell, and so those are two differences. Both of these are used in movement, but the cilia is used more to move materials around the cell, and the flagella is used more to move the cell itself. Okay, so if we talk about how the organelles structure themselves, and specifically how the cytoskeleton aids that support within a cell, there are protein fibers, and there's three of them that we're going to talk about in this class. One of them is microfilaments, one is intermediate filaments, and one is microtubules, and they range in size from the smallest as a microfilament to the middle, intermediate filaments, to the largest, microtubules. And all three of these different sized proteins are going to add structure to our cell, much like scaffolding, or much like that picture of the house where you have different sizes of boards and different sizes of lumber that all aid in the structure and the support of that house. And so if we go back to that picture, notice, if I can get my pen, that this layer right here is much thicker than these, and even these on this side are much thicker than these that fit right between the joists, or right between the 2x4s that make up the wall. So these corners are much thicker than, like I said, these. And so you can think about this in terms of three sizes. One, you have this thickness, biggest. Two, you have the corners, which are kind of the intermediate, and then the smallest, and they each don't have to support the same amount of loads, but they all have their own specific place to provide the cell or house with the structure it needs in order to live and move like a cell needs to. Okay, so these cytoskeleton components, both or all three, the microfilaments, intermediate filaments, and microtubules, are going to provide structure similar to scaffolding, and they're going to allow organisms to move. And one organism in particular, the amoeba, is a single cellular eukaryote, meaning it has a nucleus, it has all the organelles we've been discussing, and it uses these protein fibers, all three of them, to move itself, and it's going to basically build projections, and it's going to reach out into its environment, and it's going to grab the environment, and then it's going to kind of pull itself along. And they don't move fast, but they move very very well within the environment they live in. Okay, they use proteins. They use these cytoskeleton projections in order to move themselves through their environment. And one thing that the cytoskeleton does for the cell is that it gives the cell its shape. Just like that picture of the house, the framing is the shape of the house. You don't just put or frame a box and the rest of the house takes shape. You have to frame the house exactly the shape you want it, and the cell is the same way. Okay, so we're going to give the cell its shape and strength based on its cytoskeleton components. Microfilaments, we talk specifically about this one. Remember, this one is the smallest, and you can remember the smallest by micro. It's the only one with micro in the name. Now, I know there's two different filaments. You have microfilament and intermediate filament, but just based on prefixes, you know that, or I would hope you know that micro is a smaller prefix than intermediate. And so microfilaments are the smallest. They're made up of the protein actin, and anytime you have a filament, both intermediate filament and microfilament, they're going to be made of the protein actin. When you have a microtubule, it's made of the protein tubulin, and we'll get to that in a minute. But the microfilaments are the smallest filament, the smallest protein, and it adds the framework that supports the cell. It gives the structural integrity between the larger, more robust proteins in the microtubules. Okay. These microfilaments are going to help the cells move by assembly and disassembly, so they're going to constantly be built up and constantly be torn down. And if you think about a cell, if I just kind of draw a cell here, if it builds this protein, and I'm kind of just building this protein as we go, and eventually it gets to the cell membrane, and then it keeps building. Well, when it builds into this cell membrane, it's actually going to cause this cell membrane to bulge, and it's going to keep building and keep building and keep building, and eventually the cell membrane is going to keep bulging, and eventually we are pushing the cell this way. If you've ever been anywhere where one of those big blow-up balls where you can get into is, or whether you've been in one or have seen one, it works the same way. Okay, you're in a big blow-up ball, and you can actually move the ball by pushing in the direction you want to go. And as you push on the cell, it rolls, or as you push on the ball, it rolls. And these microfilaments are the same way. Now, as we're building this way and pushing the cell this way, the proteins back here obviously have to disassemble, and so if we're building this way, we are going to basically disassemble these as we go, and our overall direction will be, like I said, in this way. All right, so these microfilaments are going to build up, they're going to push the cell along, and it's going to produce a net movement in one direction. Okay, and these are very important for muscle cells because they're going to contract and relax. And muscle fibers are long, multinucleated cells, and they're very good at moving organisms. You wouldn't be able to move without the cells or without the muscle cells, but specifically they wouldn't contract or fire or move you without this protein microfilament and the assembly and disassembly. Microtubules, these are the largest proteins. Again, tubules made of the protein tubulin, and the reason it's called a tubule, and I know micro means small, and you're probably thinking, why is the largest protein use a micro prefix? And the answer is these proteins are all small, but this is the biggest of the small proteins. And filaments, both the intermediate and microfilament, are a solid tube or a solid filament. That's why they're called filaments. These are tubes, and so when you think about a tube, there is a hollow straw-like protein, and not only are these the biggest, but they are hollow, and they are the ones that maintain the cell shape. And so if you think about the picture again that we saw at the very beginning, these microtubules would be the corners of the house. They would be the floors of the house. They would be the roof lines of the house. They would be the thing that gives the house its shape, and they are the pieces of that house framing that support the most weight. And they, like I said, have to be the largest because they are supporting the cell and maintaining that shape. These are the proteins that are going to help build projections from the cell, and that's when we start getting into the cilia and flagella. And so you need, again, the strongest, largest proteins to help move the cell or move things around the cell. Here's a picture showing all the different types of filaments and all the different proteins. Notice that this one right here, these yellow ones, see how that one's open on the end? This is the microtubule. See another one here, and they are not flowing everywhere. They are literally just the protein that gives the cell its shape, and they are the largest protein that all these other smaller proteins kind of are hooked to. The intermediate filaments are these pink ones, and they're the ones that are next in size. Again, they are solid. They are not hollow. They are a filament. They are composed of actin, whereas the tubule is composed of tubulin. And then you have the smallest ones, all of these ones right here, which are going to give the cell basically all of the structural integrity. These are the microfilaments. These are the smallest ones, and these are the ones that are going to be building up and breaking down to move the cell. Notice that this is a vesicle, and we're going to see a video in class, but the vesicles actually use these microtubules kind of like a cellular highway. Again, these are the biggest protein, and so we have a motor or carrier protein that is actually going to carry these vesicles down the length of these microtubules, kind of like a delivery truck on a highway. And we're going to use these microtubule highways that extend from the rough ER and travel. These vesicles are going to travel from the rough ER to the Golgi, and then from the Golgi everywhere else. And so don't think of these vesicles as kind of these membrane-bound bubbles that just kind of float around. They actually have to travel a route, and those routes are determined much by the microtubules within the cell. Notice that these little dots here that are on the rough ER make the rough ER appear rough, and so those would be the ribosomes, and there are ribosomes here. And remember in class we talked about the ribosomes not only can be attached to the rough ER, which gives it that rough appearance, but the ribosomes can also be attached to the cytoskeleton, and these are the ribosomes we say are free in the cytoplasm. Again, free doesn't mean that they're just floating around. Everything within the cell has to be kind of anchored, and these organelles are anchored to the cytoskeleton to give it something to attach to. So, if we talk a little bit about movement and specifically cilia, the cilia is used to move materials past the cell. And remember, if we have a cell and we have all these little hair-like projections, and I'm not going to make them all, but let's say we have this food item and we want to move this food item this way. Well, we're going to basically hand off kind of like a bucket brigade, and eventually we'll get that food item all the way into where the cell wants it to be. And so the cilia are good at moving materials past the cell, whereas flagella are used to actually move the cell. Okay, let's just assume that this line is the flagella. The cilia are moving the materials. The flagella is actually going to move the cell. Some cells use cilia to move in their environment, but the majority of cells are going to use the cilia to move materials past. Those cilia are tiny hair-like projections that cover the exterior of a cell, and they specifically are made of dining arms, and they bend, causing, usually with the use of ATP, but they cause the neighboring cells to bend as well, and that's why I made that that analogy of the wave in a sports stadium, because as one person stands up and goes nuts and does the wave, then the next person does it, and the next person, and the next person, and then that's how we get that whole wave-like motion going, and it creates that waving motion. ATP is required to move the dining arms. It is an active process. It doesn't just do it for the fun of it. It actually requires energy, and that energy is in the form of ATP. So if we look at these cilia, and the cilia are produced by dining arms, and they have these microtubule doublets, and so when we add ATP, one of them is basically going to bend away, and the other one's going to kind of slide past it, and because there's this sliding force, it's going to cause a whip-like motion, or a wave-like motion, to happen where one triggers the next, which triggers the next, which triggers the next, which triggers the next, and creates that wave-like motion. Flagella. Remember, flagella is the big, or in sometimes multiple tails that move the cell around its environment. And there's generally one or two, but there can be multiple. And the flagella is made up of 8 triplets of protein microtubules. And like I said, the flagella is used and is very good at moving itself through the environment, or moving the cell through the environment. It's very strong. Again, it uses ATP. It's going to use the same kind of microtubules, the same kind of protein that the cilia use. And here's a picture. This is an actual electron micrograph cross-section. And so you can see these microtubule doublets. There are 8, I believe, or 9 actual doublets, and one in the middle. And so all of these ones still have the dining arms like they did in the cilia, exactly the same proteins. This one is just much elongated, and it creates that tail, and it's going to whip through its environment like a snake swimming in the water to propel the cell in any direction it wants. Okay, and the very last thing we're going to talk about, and I know I talked about it I think 2 lectures ago, but I'm just going to kind of repeat the cell wall. Remember, the cell wall is a rigid layer on the outside of the cell membrane. The cell membrane comes first, cell wall's on the outside. The cell wall is found in plants, fungus, and bacteria. Bacteria are going to have a cell wall that is composed of peptidoglycan. Plants are going to have a cell wall that's composed of cellulose, and fungus is going to have a cell wall that's composed of chitin. All 3 of those molecules are complex polysaccharides, and if you remember back from chapter 2, we talked about what a polysaccharide is and how a polysaccharide is formed, and it's just a repeat of that whole structure dictates function thing, where structure is directly dependent on its function, and the function is directly dependent on the structure. Okay, something has to be structured right in order to function correctly, and all 3 of these cell walls are different because they're made of different things, which is why plants look different than fungus, and fungus acts different than plants, and bacteria have a cell wall that looks much different than plants and fungus. But at the end of the day, all 3 of these types of cells have cell walls, and it causes the cells, again, to be compartmentalized, meaning individual cells can either live by themselves or they can produce multicellular organisms, and these membranes are composed of phospholipids, but the cell wall is made up of the polysaccharide, the complex carbohydrates that give structure and support and protection to the cell. I think that's going to do it for today. Mr. P. See ya.